Hi, student. Good morning. Today we are going to discuss the topic number 4 of IGCSE biology. And in topic 4, we have to discuss about the biological molecule, especially macromolecule and the micromolecule. In macromolecule, we have to discuss about carbohydrates, protein, and fats. Okay, and also we have to discuss, some type of micromolecule, especially like amino acids. Okay, and RNA, DNA. Okay, we will also discuss water molecules. So let's to start. Now see here, a biological molecule. See, a nutrient is a substance which is needed for growth, repair, and metabolism. The three main nutrients are, okay, actually these nutrients, are found, okay, in our food, okay, and they are macromolecule, the large molecule, specifically carbohydrates, proteins, lipids, or fats. So the first thing that we should know that which elements make carbohydrates, protein, and fats. Okay, and what are the differences, and what is the similarity? Okay, in all. So see, carbohydrate. Carbohydrate contains carbon, hydrogen, and oxygen, but nitrogen is not found in carbohydrate. Lipids, it contains carbon, hydrogen, oxygen. Also, nitrogen is not there. Okay, and protein. In protein, all like four elements, including nitrogen, is there. So this is the like the elements. Which elements are found? Okay, carbohydrates we may be simple, soluble sugar or complex material like starch and cellulose. All contains carbon, hydrogen, and oxygen. Means if you are going to study any carbohydrate, so carbohydrate is the name of three elements: carbon, hydrogen, oxygen. For example, the formula of simple glucose is C6H12O6. This glucose is also called as a simple sugar. Okay, so there are like C6H12O6. Now there is a like types of carbohydrates like monosaccharide sugar with a single carbon ring, for example glucose and fructose. And disaccharides means sugar with two carbon rings, for example maltose and sucrose. Means in disaccharide, for example, like when glucose combine with glucose, so it make maltose. Okay, so means two monosaccharides are found in disaccharides. And polysaccharides mean many monosaccharides are found. Okay, many glucose molecules join together. For example, glycogen. Glycogen is an example of polysaccharides, which is the storing carbohydrate in animal tissues. And starch, which is also the polysaccharide, which is also found in the storing carbohydrate in plant. And cellulose, that consists of even longer chains of glucose molecules. Mono and disaccharides are readily soluble in water. Okay, they are really readily soluble in water. Polysaccharide are not readily soluble in water. Now see, protein, another the biological molecule or the macromolecule is protein. If you are going to study the structure of protein, you see here protein contribute to the structure of cells, okay, and to the cell membrane, mitochondria, ribosome, and chromosome. Okay, it makes like different structures. Okay, and enzyme, you can see here, control the chemical reaction that keeps the cell alive. Present in the cell membrane system, okay, in mitochondria, in special vacuoles, and in the fruit part of the cytoplasm. So actually here, this is the importance of protein, that protein is making like the different structures. For example, mitochondria, ribosome, and chromosome. And protein also responsible to make enzymes. Okay, so protein are large molecule made up from small units called as amino acids. So protein is a polypeptide chain of amino acids. Now the fats or lipids. Okay, see what is the difference between the fats and lipids, or the fats and oils. So oils are liquid at room temperature, and fats are solid at room temperature. So fats and oil or lipids. Okay, so lipids are liquid. So lipid are large molecule made from a small units of fatty acids and glycerols. Okay, now we are going to discuss about the food test. Okay, so see the first food test, test for the starch. A starch is detected using iodine solution. Yes, if you are going to just put 1 or 2 drops of iodine, okay, the color of iodine turns into blue black, because the normal color of iodine, is brownish red. Okay, so if there is a presence of a starch in any sample of food, so the color of iodine turns into blue or black. And another, test for reducing sugar. Reducing sugars are detected using Benedict solution. Okay, using Benedict solution. The solution will change from clear blue to cloudy green. Okay, the yellow, and finally the red. Finally, to a red precipitate of copper oxide. So see, reducing sugar test. This is also called as Benedict test. Why? Because the Benedict solution is used. Normal color of Benedict solution, is blue. But if there is a presence of reducing sugar in a food sample, so it turns, finally it turns into brick red color. Now, test for protein. Protein are detected using the Bayreuth reagent. This turns purple color when mixed with the protein sample. Means the normal color of Bayreuth reagent, is blue. Okay, and if there is a presence of protein in any food sample, so it turns into purple. So this test is also called as the Bayreuth test. Test for lipids. Lipids are detected using the emulsion test. Okay, and what is this test? The test substance is mixed with 5 cubic centimeter of ethanol. Okay, for example, if you are having the sample of oil or fats, okay, you put it into the test tube and then adds about 5 cubic centimeter of ethanol or ethyl alcohol. So an equal volume of distilled water is also added. Okay, so if there is a presence of oil or fats or lipids, so you will observe that a milky white emulsion forms if the test substance contain lipids. Now, test for vitamin C. Okay, this is also called as DCPIP test. Okay, and for this test, we have to use the chemical that is DCPIP. Okay, so see, add up to 2 cubic centimeter of fresh lemon juice into the plastic syringe. Add into a test tube with 4 cubic centimeter of 0.1% solution of DCPIP, a blue dye. You see the color of this, this reagent is blue. The DCPIP will suddenly becomes colorless as the juice is added. Note the amount of juice is added. Okay, now in the repeat number 3, repeat the experiment but with orange juice in syringe. If it takes more orange juice to decolorize the DCPIP, the orange juice must contain less vitamin C. So this is, this is vitamin C test. Okay, and it is also called as DCPIP test. Okay, because we are using DCPIP reagent or dye. Okay, which is normally color, which is normally like blue color, and when it combined with a vitamin C that is found in the orange juice, it turns to colorless. Now after that, we will discuss about like the proteins. Okay, there are about uh, protein and amino acids. So there are about 20 different amino acids in animal protein, including alanine, leucine, valine, glutamine, cysteine, glycine, and lysine. A small protein molecules might be made from the gene consisting of 100 or so amino acid. Each type of protein has its amino acid arranged in a particular sequence. The chain of amino acids in the protein takes up particular shapes as a result of cross-linkage. Cross-linkage formed between the amino acids that are not neighbors. The shape and the structure of the protein molecule has a very important effects on its reactions with substances. For example, active site of enzyme, and the binding site of the antibodies. If you study like the enzyme structures, you will see that there is a one site that is known as the active site where the substrate is bind. Okay, and when the substrate binds, so it is called as the lock and key model. Okay, according to which the substrate is to be broken down. Okay, when it's going to be fixed into the structure of the active site. Now, binding site of antibodies, see, also it is found in the binding site of antibodies. Now, see, denature, yes, you know that all sort of protein, especially enzyme, okay, they work on a specific temperature, okay, and their optimum temperature, if you just study the human beings, okay, their optimum temperature, is 37 centigrade. If the temperature is to be increased, okay, when a protein is heated over 50 centigrade, the cross linkage in its molecules breaks down and the protein molecule lose its shape and will not usually regain its E1 when cooled. Yes, for example, the egg white, if you are going to just give a high temperature to the egg white, so it's going to shrink. Now come to the next one. See here, this one about the DNA. Now, if you are going to study the DNA, you will see that in the diagram you can see, you will see that the DNA, is a double-stranded molecules, okay, and which contains so many nucleotides. Now, what the nucleotide is? See, one nucleotide contains deoxyribose sugar, one nitrogen base, and phosphate group. Okay, and when so many nucleotides combine together, they make a polynucleotide. And when two polynucleotide chains coiled with each other, so it makes the it makes the DNA structures. Okay, so now you can see made of long chain of a nucleotide forming into two strands. Okay, a nucleotide is a carbon sugar molecule joined to a phosphate sugar. Okay, so you can see here deoxyribose sugar is there. Okay, nitrogen bases, there are four types of nitrogen bases are there. For example, adenine, thymine, cytosine, and guanine. Okay, so the nucleotides are joined by their phosphate groups to form a long chain. So you can see here these nitrogen bases are combined with a deoxyribose sugar, and on the other end of the deoxyribose sugar is combined with phosphate group. So this is one nucleotide, and so many nucleotides are there. A phosphate and sugar molecules are the same all the way down the chain, but the bases may be any one of the four listed. The DNA in this chromosome consists of two strands that chain of the nucleotide held together by chemical bonds between the bases. The size of the molecules ensure that A means adenine, always pair with T means thymine, and cytosine pair with guanine. The double strand is twisted to form a helix. Okay, so this double strand, twisted to form a helix. As I've told you that the two polynucleotide chains, are twisted, okay, and they make a helix, okay, or the double-stranded molecule that is DNA. Now, after that, the water. This is the last, topic of topic number 4. Okay, that is about the biological molecules. Water, you know very well that most of the cells contain about 75% water. Okay, and will die if their water content falls much below this. I mean, even in some religious book, it is said that each and everything is made by water. Okay, so if there is a water, there is a life. And if you are going to study the cytoplasm, protoplasm, what is cytoplasm, protoplasm? Okay, it's also water, which with having some other organic and inorganic substances, but mostly water is there. So if there is a water, there is a life. Okay, what is good solvent? Okay, and many substances move about the cell in water solution. So water is considered a universal solvent. Why? Because it has the capability to dissolve many substances. In guinea plant, water combines with CO2 to form sugar. In animals, water helps to break down and digest food molecules. Blood is made up of cells and liquid called plasma. Yes, see, blood also contain, plasma. This is about like, plasma contain 92% of water and act as a transport medium. Means water has, and this plasma has the capability to dissolve many substances. Okay, it like the urea is dissolved in plasma, glucose is dissolved in plasma. Okay, and if the urea has to be expelled out from the body, so this blood plasma take urea to the towards the kidney, from where it is to be expelled out in the form of urine. And also blood plasma transport glucose, okay, and take glucose, okay, or transport glucose to the each and every cell, where like the glucose react with oxygen makes energy. So water performing so many jobs. Okay, water also act as a transport medium in plants. Water passes up the plant from the roots to the leaves in xylem vessel and carries with a dissolved mineral ions. Phloem vessels transport sugars and amino acids in solution from leaves to their places for of use or storage. Water plays an important role in excretion in animals. Yes, as I've told you that urea expel out from the body. Why? Because the urea is dissolved in blood plasma. It acts as a powerful solvent of excretory material. Water has a diluting effect, reducing the toxicity if the excretory material is. So water, without water, there is no life. Without water, there is no transportation. Without water, there is no formation of energy in the body. Okay, so water, is very important and essential for life. Okay, if there is a water, there is a life. If there is no water, there is no life. Okay, so thank you very much. Okay, so if you have any question, you can ask me. Okay, my WhatsApp number is given here, my email address also there, and soon I'm going to just upload this lecture on the YouTube and TikTok. Okay, so if you need any question, if you need anything, okay, you can also write down the comments that like what do you require. Okay, if you want a special like lecture any topic, okay, you can also write it down, okay, in the comments of YouTube channels or the TikTok. Okay, okay, thank you very much. Bye, take care.